Good day everyone. We are pleased to present our laboratory report for the Tissue Culture Technology course. My name is Gunavadi Maniam and I am joined by my group members Sujita Suraj, Renisha Govindasamy, Siti Nur Sazlin binti Muhammad Zamri and Vishalini Shanmugam. We have worked collaboratively on this project and we are excited to share our findings with you. Our presentation today will cover the topic of plant tissue culture, specifically focusing on the multiplication or subculture. We have structured our report to provide a comprehensive overview of our introduction objective, material and methods, results, discussion and conclusions.
My name is Rita Suraj and I will explain about the introduction. So first, what is plant tissue culture? Plant tissue culture is a collection of techniques used to maintain or grow plant cells, tissues or organs under a sterile condition on a neutral culture medium. It is a crucial method in plant biotechnology and serves various purposes, including plant propagation, genetic modification and conservation. Haberlin was the first scientist to produce whole plants from the plant tissues, and so he is popularly called as father of tissue culture too. So there are various types of plant tissue culture. Here are some of the examples such as embryo culture, organ culture, callus culture and cell culture too. Next, how to perform the plant tissue culture. The process will start with initiation phase where at this stage the tissue is initiated into the culture. The tissue of interest is obtained, introduced and sterilized to prevent the process from any contamination. This process is followed by the multiplication phase where in this stage the sterilized excipient is introduced into the medium composed of growth regulators and appropriate nutrients. They are responsible for the multiplication of cells. This undifferentiated mass of cells is known as callus. For the third, it will be root formation, where the roots start forming and the plant growth hormones are added to initiate the root formation. Consequently, we obtain a complete plantlets too. Next, it will be shoot formation, where the plant growth hormones for the formation of shoot are added and the growth observed for a week. And last, the process will end with acclimatization, where the plant starts developing, it is transferred to the greenhouse to develop under controlled environmental conditions. It is finally transferred to the nurseries to grow under natural environmental conditions. Now, let's talk about two main things that plays a major role in plant tissue culture. The first one is multiplication. Multiplication is where the micropropagation will occur. Micropropagation is a plant propagation using extremely small pieces of plant tissue taken from a carefully chosen and prepared mother plant and growing this under a laboratory condition to produce new plants. A protective hood to allow work on a plant material in clean condition is a need to do this process. Meanwhile, subculturing is the removal of the medium and transfer the shoots or callus from the previous culture into a fresh growth medium, a procedure that enables the further propagation of the cell line or cell strain occur. So at the end of the day, we will produce a disease-free and healthy plant. Shortly, we can say that multiplication occurs when small plant tissue produce large number of identical plants. Now, let's talk about the second main thing, which is acclimatization, which is also known as hardening of process. It is a process where we will adapt the in vitro plants to ex vitro condition. This is where we will do transplanting, and it's a crucial process where moving plants, particularly young seedlings or house plants, from a controlled indoor environment to the harsher, more variable condition outdoors will occur. This gradual process helps plants to adjust to changes in temperature, light, wind, reducing the risk of transplant shock and improving their chances of survival and growth.
The first objective is to multiply the number of plantlets. Our second objective is to subculture the plantlet into fresh medium to accelerate their growth. These objectives guided our experimental procedure throughout the project.
Okay, this slide is about material parts. For the banana subculture, we have plant culture, MS medium, 70% ethanol spray, screw capped jar, forceps, scalpel and blades, and also petri dish. For the acclimatization, we have subculture plant, which is orchid plant, plastic cup, plastic container, hole puncher, cocoa pits, and also tissue. For the methodology part, this flowchart states for the banana subculture. First, a banana culture that was suitable for subcultures that were crowded or tall enough was chosen. The unnecessary shoots and roots were trimmed and removed. Explants were separated into individual plantlets aseptically. Then, shoots about 3 cm long were cut off and transferred to MS basal medium. Then 2 or 3 explants were placed into a culture jar that contained fresh medium. Then the plants were gently pressed down with forceps slightly into the medium and the jar was closed using a jar lid. The culture were kept in a controlled growth room at temperatures of 25 degrees for 2 weeks. Then the culture was observed for 4 consecutive weeks, and the number of plants obtained and the contamination of the end of the 4 weeks was recorded. Okay, next, this flowchart states for acclimatization. The plant subculture was removed from the jar and cleaned thoroughly. Then a tissue was weighted in a plastic container, and the clean plant was placed into the plastic container, then leave it for 2 days with the lid slightly open. After 2 days, the plants left in the plastic container were transferred to a plastic cup filled with a cocoa peat to be placed in the nursery. After that, plant was sprayed with water once every 2 days or every week until the plant grew.
As we can see, this is the result of the subcultured plant. On the first picture, we can see that the plantlets were subcultured into the medium agar in the jar. So the second picture is the plant that grew from the jar for 2 weeks. So we transferred the plantlets to a sterile box. This is the first stage of the acclimatization phase. So we can see there is a bit of root development and also leaf development. Discussion for subculture. After transferring the plantlet to the sterile box, their immediate response was observed. Healthy plantlets maintain a green coloration and turgor, indicating minimal stress. During the first week, the plantlets were monitored daily. Healthy plantlets showed sign of new leaf and root growth, with new leaves appearing green and vibrant, indicating effective photosynthesis, and new roots developing, essential for nutrition uptake. By the end of the observation period, successful subculturing was confirmed by the continued growth of new leaves and roots without sign of contamination or stress. This is the result for the acclimatization. On the first picture, we can see that there is developed root and also leaves. So the second and third picture shows the final stage of the acclimatization. That is, after the mid stage, we took the plant and transferred it to the nursery. So we can see the developed plant and also the roots there. Discussion for acclimatization. During the acclimatization phase, plantlet were gradually exposed to external conditions by reducing humidity and increasing light intensity. In the initial phase, this environmental adjustment helped the plantlets adapt without experiencing shock. Daily monitoring focused on identifying stress signs such as wilting, leaf drop, or discoloration. In the mid-acclimatization phase, growth parameters including the size and number of new leaves and the length and density of new roots were recorded. Healthy plantlets showed continuous growth with new leaves and roots developing robustly. During the final phase, plantlets were transformed from the controlled environment chamber to a nursery. They were gradually exposed to ambient conditions over a period of days to weeks. Final assessment of the plantlets included evaluating their overall health and robustness with successful acclimatization indicated by a strong root system, multiple healthy leaves, and the absence of stress symptoms.
In the conclusion, subculture and acclimatization is an important component for plant tissue culture. Subculture is the process of transferring tissue culture to fresh growth medium. It is necessary to keep the tissue culture plant in a healthy developing state and allow the plant to thrive in sterile and controlled environment. This method promotes the multiplication of plantlet, providing a steady supply of the plant that are disease-free and genetically informed. Next is the acclimatization. This acclimatization is the adaptation of the organism to a new environment. When tissue culture plants are transferred from the lab to soil, they are exposed to abiotic stress like altered temperature, light intensity, and humidity condition, and biotic stress like soil. It is important to plant to develop their structure and functional system like root and increase their residency of the pathogeny, which lead them to a better growth and development. To ensure this, some of the method must be emphasized properly. The first method is preparation of the media in using a better and healthy explant. It is important to choose an appropriate culture medium and transferring using a better and healthy explant. Next is sterilized environment. It is important to make sure the environment are micro-free and sterilized properly along with the tools which are using for culture medium. Next is environmental control. During the acclimatization, it is important to the plant provide the natural optimum environment, which is the better humidity, better lighting, and the optimum condition of the temperature. Last but not least, observation. It is important to monitor the subculture and the acclimatizations of the plant. During their time, we must ensure their process of the growth and avoiding any type of contamination during the growth time. That's all. Thank you.